Hello and welcome to our session today. In this presentation, I will discuss the optimization of serum-free in vitro culture conditions for genome editing CD34 cells. My talk today will focus on three main areas, including the importance of cell culture when genome editing hematopoietic cells, combining these conditions to create a standardized workflow, and how cell culture assays used in the lab may be transitioned to cell therapy manufacturing techniques as you move to clinical trials. First, a quick introduction to myself. My name is Amanda Fentiman. I'm a product manager at STEMCELL Technologies. Shown here is myself and my dog Penny, who is thoroughly enjoying this change to working from home. Prior to joining STEMCELL about 5.5 years ago, I worked in a lab at the BC Cancer Agency here in Vancouver, studying developmental hematopoiesis. Okay, so knowing that we're here at ISSCR, a conference bringing together researchers from all areas of stem cell biology, I will start with a little bit of background on the field of hematology. One thing that I find really sets HSCs apart from other stem cells is their long history of use in the clinic. So several years before they were described in the papers shown on the slide here by Drs. Till and McCulloch, the first bone marrow transplant was actually already taking place in Seattle by Dr. E. Donald Thomas. Much of the early work that pushed these cells to the clinic before we even understood their biology was a result of attempts to treat radiation-induced cytopenias that resulted after World War II. Since then, we have learned a lot more about the heterogeneity of hematopoietic tissues, much of which is actually still being elucidated today and presented at meetings such as ISSCR. One thing that hasn't changed drastically, though, from these early days is that similar to how HSCs were first used, they are still defined largely by their function. To explain what I mean by that, I have two brief definitions on this slide. First, hematopoietic stem cells, or HSCs. This is a term which refers to the population of stem cells capable of long-term multi-lineage differentiation and self-renewal in vivo. There is no one single marker to pick these HSCs out of a population such as bone marrow. And conversely, hematopoietic progenitor cells, or HPCs, is a term referring to a population of more lineage-restricted cells, which exhibit high proliferative capacity, but cannot self-renew like a true hematopoietic stem cell. While advances are being made in the determining improved definitions of phenotypically described populations, hematopoietic stem and progenitor cells are traditionally differentiated from each other by their functionality in vivo, and including engraftment experiments. Therefore, when discussing the broader, more heterogeneous population, I'll use the term hematopoietic stem and progenitor cells, or alternatively, referring to these cells as CD34 cells due to their shared cell surface marker that is expressed by human hematopoietic cells. While we'll talk about more recent research, it's important to remember that traditional hematopoietic stem cell transplants have actually been used, as I said, for over 50 years, and more than 50,000 transplants are carried out annually across the globe, as this is a great method to treat disorders such as leukemia and sickle cell disease. These transplants are made up largely of bone marrow and more recently sources such as peripheral blood. These traditional techniques employ large sample sizes and are isolated from adult donors, so they are often not restricted by cell number. The cells themselves do not require genetic manipulation by nature. However, there are issues around HLA matching to ensure transplant success. You may be asking, if we have these standard treatments already, why are we still working to develop new cell therapies with hematopoietic cells? And the thing here to say is that these transplants, while they've been used for a long time, are not perfect. And there are still issues in finding suitable matches, particularly for more genetically diverse or underrepresented populations, and for developing methods that will help prevent some extremely unwanted events post-transplant, such as graft failure or graft versus host disease. As you can see in the graph on the right, the blue bars at the bottom show the prevalence of bone marrow as a transplant source, and the yellowy green bars in the middle represent peripheral blood as a transplant source. Since about the year 2001, you can see the growing orange bars on the top, which represent an increase in the use of cord blood stem cells for transplant. However, with the benefits of cord blood-derived HSCs, including things such as less stringency for HLA matching, there of course are challenges related to the small sample size and therefore HSC cell numbers, their quality, and their potency. So for new research, there's a lot of groups looking at finding new solutions, many of which involve the culture and modification of these cells. So in vitro cell culture, as I said, can be an important tool when developing these new hematopoietic cell therapies, either stimulating or priming peripheral blood-derived CD34 cells prior to genetic manipulation, or driving the expansion of cord blood HSCs to improve engraftment and recovery post-transplant. Cell culture is a way that people are looking at improving these processes for both of these cell sources. When CD34 cells are isolated from a patient's own peripheral blood, after mobilization with drugs such as GCSF, they can then be modified and transplanted back into the patient in what is termed an autologous transplant. When cord blood CD34 cells taken from a public bank, for example, are treated to expand HSCs and transfused back into an unrelated donor, this would be an allogeneic transplant. Other techniques which rely on cell culture include in vitro methods for influencing the differentiation potential of hematopoietic stem and progenitor cells, generating mature cells for transfusion or immunotherapeutics. Many of these applications are based on research that began several decades ago and include translational studies that are either actively moving towards the clinic or already being investigated currently. And while HSC expansion sounds relatively straightforward as a method to overcome the rarity of these important cells, this technique can have some trade-offs regarding cell function. So the numbers at the top of the slide here show the HSC fraction of cord blood and bone marrow as an example, which as you can see is quite low among the entire mononuclear cell population. Unlike pluripotent stem cells, we cannot, unfortunately, generate 100% pure, large numbers of clonally expanded populations of HSCs or even grow them in culture for extended periods of time beyond about 2 weeks or so. Part of these intricacies are due to where HSCs are in the natural environment. So they're located in the bone marrow niche. When HSCs are out of this protected niche, they are exposed to cytokines or other signals which may cause them to divide quickly and lose their stemness. Essentially, as we cannot yet perfectly approximate the niche in vitro, all hematopoietic stem cells in culture do undergo some maturation as they proliferate. So when looking to expand HSCs, the key is really balancing this maintenance of their really important attributes such as stemness and maintaining this potential for later engraftment, but also applying any modifications or differentiation that is desired. So one area of research that has been quite active over the past decade or so is the quest to find a perfect small molecule or other extrinsic factor that can help approximate niche effects in vitro while allowing HSCs to maintain stemness and expanding or making them at least more amenable to modification. Shown here on this slide are a few of these papers, including the identification of aryl hydrocarbon receptor SR1 by Dr. Boitano, and another paper with work from Dr. Colleen Delaney at Fred Hutch looking at the effects of Notch ligand stimulation on progenitor cell expansion, and Dr. Fers from the Université de Montréal identifying UM171 and 729. Several of these molecules have been investigated clinically in the past or are currently being investigated. So following this brief kind of crash course in HSC biology and some of the research that is pushing this field forward, let's take a specific look at genome editing, so a technique that has been used fairly recently on hematopoietic stem cells. So as those of you that are experienced in genome editing are likely well aware, in vitro culture of target cells is at least minimally required to stimulate them, transduce them, or electroporate them. Although high levels of cell expansion are definitely not a requirement in this case, cells must at the very least be healthy and active to ensure success. This is of utmost importance, particularly when working with tricky primary cells such as HSCs, and especially when using disruptive technologies such as electroporation to deliver protein complexes into the cell. So on this slide as well, you can see a couple recent publications from Dr. Mark DeWitt in Jacob Korn's lab and Dr. Daniel Deaver from the Porteous lab at Stanford. These groups both were some of the first papers to use CRISPR-Cas9 mediated editing of human hematopoietic cells, and they used conditions in these papers that were traditionally used for stimulating high levels of cell expansion, but in this case used them for shorter term cultures required for genome editing. Post-editing, cell culture-based assays such as a CFU assay or even just in vitro differentiation were then used in several of these papers to evaluate any changes to progenitor cell function in vitro. Efficiency of gene knockout or repair can also be determined through use of assays such as the CFU assay. And so just to point out, one thing that really started to get us so excited was that in some of these first papers shown below, both of these groups used our STEMCELL FEM2 media with cytokines added, and then later methicult media for CFU assays to characterize their edited cells. So as I said, when we were watching these papers come out, we were getting really excited because we soon realized that really researchers out there were telling us that our media that we knew was really good for CD34 expansion was also optimal for CRISPR-mediated genome editing of these cells. So this led our scientists at STEMCELL to take things further and testing an extended workflow of more of our tools kind of built into this diagram you see here, including products for cell isolation of CD34 cells with EasySep, our serum and animal component-free media formulations for this context, expansion supplements, and then added to that are some of our newer guide RNAs and molecular biology tools to actually modify these cord blood-derived cells. So let's just focus a bit more specifically on these pre- and post-electroporation culture steps shown here. So in short, the protocol that we worked out for editing CD34 cells involved culture for 48 hours pre-electroporation with STEMCELL media, our CD34 expansion supplement cytokine bullet, and the small molecule UM729. The group of these products is shown there on the right. Post-electroporation, culturing for an additional 96 hours allowed Cas9 to knock out our target genes, after which time cells were analyzed by flow cytometry and plated in CFU assays for an additional 14 days. To some people, it might seem odd to focus so much of this talk on just the culture conditions, as this is usually the detail that is left to the supplemental section of a paper. However, at STEMCELL, we're very passionate about making sure scientists have the right culture conditions for which to really build their new technologies upon when working with these cells. So in the development of these protocols, we performed a few case studies, including knockout of the beta-2 microglobulin gene, which encodes for the MHC class I protein. This target was chosen as it can be read out by staining for this as a cell surface marker and analyzing by flow cytometry. In the panel on the left there, you can see that when large cell numbers are desired, the first orange bar, representing SFEM2, generates the largest total number of CD34 cells after the 4 days post-electroporation. With animal component-free STEMCELL ACF media, the second orange bar there, not far behind in terms of cell numbers generated. In the panel on the right, you can see that the more primitive CD34 positive, CD90 positive, CD45 RA negative population in particular shares the same trend with the relative frequencies of edited and non-edited cells shown. Overall efficiencies of editing were actually quite similar in our media as well as competitor media that we compared to. However, this was partially standardized as we used our STEMCELL CD34 expansion supplement in all conditions and as well added UM729 to really boost up that expansion. So after confirming that these conditions do allow for high performance culture and modification of more primitive CD34 positive cell subsets, Dr. Leon Lin and our team looked at even further refining which cells were actually growing and being maintained in culture and subdivided this population on the basis of CD34 expression level. So shown here are cell phenotyping and fold expansion data from cord blood-derived CD34 cells cultured for 7 days in SFEM2, STEMCELL ACF, or other commercial medium, again using our CD34 expansion supplement for cytokine source and UM729. So even after this additional 1 day, so 1 day more than the gene editing protocol employs, this full 7 days of culture, we can still maintain a healthy population of CD34 bright, CD90 positive, CD45 RA negative cells. In the graph on the right, our BSA-containing medium SFEM2 provided the highest fold expansion per input cell, and our animal component-free STEMCELL ACF, which contains only recombinant sources of protein, was found to meet or exceed competitors that were serum-free or xeno-free themselves. This same pattern again, not shown here, but is maintained when we look at the bulk CD34 cell population expansion, achieving 40 times and 20 times expansion per input CD34 cell in SFEM2 and ACF, respectively. So to look a little bit at the function of these cells, next, Dr. Lin sorted cells after 7 days of culture based on their CD34 expression intensity. CFU assays were then performed to assess the progenitor cell potential at each expression level of CD34. On the left shows the FACS plot with gates used for sorting each population, and the viable cell gate, not shown, was used as well, of course. So sorted cells were plated in methical optimum and cultured for 14 days in a smart dish plate before imaging and analyzing on STEM vision. This data supports that even after 7 days of culture, the progenitors are capable of forming colonies. They're still there in culture and are highest in frequency in the CD34 bright population. Using similar experiments after 7 days of culture in those same conditions, cells were instead plated into a 96 well plate with methicult medium for single-cell CFU assays. You can see here the CD34 bright population produced colonies of all subtypes, including a single gem colony in that little orange bar at the top. Shown above each bar in the figure on the left is the colony-forming potential of cells in each population, again with the highest frequency, so about 50% of cells capable of forming colonies in CFU assays in the CD34 bright subset. On the right, total colony numbers are shown, which is also shows the highest number of colonies in the CD34 bright population, though there are colony-forming cells present in cells subtypes of all CD34 expression levels. Even when you can see between the two cord blood samples, there may be high sample variability between the donor samples. So circling back to how our conversation began here, talking about the clinic and knowing that ultimately research involving HSPCs has a high likelihood, or at least a good shot at going on to clinical trials, how do these culture conditions and assays fit into a translational context? So as was just shown in the previous data, STEMCELL ACF is one of our standardized media formulations capable and developed for HSC culture, capable of supporting 20-fold CD34 cell expansion and the maintenance of more primitive cell subsets. This medium will actually be further improved in the fall with a new version launching. It's important to note that everything I've just shown you is generated with our current phenol red containing STEMCELL ACF formulation, but all of that data is still applicable to this new medium, as the only difference that will be happening is the removal of phenol red, and also very excitingly, the manufacture of this medium to relevant 21 CFR 820 guidelines. So this is an exciting next step for us at STEMCELL, as well we currently support researchers in early stage clinical trials with our STEMCELL media. This will really help us be able to help take people from preclinical research and continue on those optimized cell culture conditions all the way to the clinic with this GMP compliant medium. So our research-focused genome editing workflow that we showed earlier, of course, also has elements that are being actively used in the development of hematopoietic cell therapies, especially that using this technology of genome editing. And the CFU assay is also able to be used in progenitor cell quantitation as you move to the clinic and need to characterize your cell therapy products and QC them while manufacturing. So for those of you not familiar with the CFU assay, or who were maybe tasked way back in undergrad in helping postdoc count their plates, this is a functional assay used to quantitate progenitor cells in a sample. This assay has a long history back from in the 1960s, when it was shown to be clonal that it can directly measure the growth and frequency of functional, viable hematopoietic progenitor cells. And really, this assay relies on the concept that one colony equals one colony forming unit, which corresponds to one progenitor cell. So setup of the assay is quite simple in its concept, and it requires simply the plating of hematopoietic cells in a semi-solid methylcellulose-based medium containing cytokines and serum, allowing the proliferation of a single progenitor while spatially isolated from other colonies. So that way, colonies may be identified separately in number, and then by the morphology exhibited, you may be able to determine the subtype of that progenitor cell, whether it is a myeloid or erythroid, etc. Having long been important for looking at hematopoietic cell samples in transplantation context, this assay has actually been shown and published to correlate strongly with successful patient engraftment following transplantation, with a couple studies on the bottom of the slide shown here from the early 2000s. So part of the continued relevance of CFU assays is, as I said, for the determination of product potency in hematopoietic cell therapy products. So when looking at, a traditional drug, that could be measured by HPLC or another analytical method. Cell therapy products are a bit trickier, as by their nature, they can suffer from higher variability and issues with lot-to-lot consistency, as they're coming from various donors of various health statuses and things like that. Regardless, before using these cells in therapeutic applications, the quality of the final product must still be defined, and both purity and potency need to be established during manufacturing. So purity, as it is, refers to what is in the product, so what cell types and in what proportion, and that may be determined most commonly by flow cytometry. And potency is defined as the ability of a compound to exert its therapeutic effects. So in this case, here we're relying on the closest connection between CFU assay results with patient engraftment. And so shown on the bottom half of the slide here, you can see an excerpt from an FDA guidance from 2014 recommending that the CFU assay be one of the assays used to determine product potency. And then this is just a couple examples here, 2 snippets from package inserts of FDA-approved non-manipulated cord blood products. They both recommend the CFU assay for QC of cells prior to infusion into a recipient. So, however, despite its power as an informative assay, I'm sure those of you that have performed this, and I have heard from others, that it's best described as more of an art than a science. So even the most expert counter, you have off days and you get tired and your eyes get sore. Therefore, it's really important to implement standardized practices when training, setting up these assays, and quantifying them. So the standardization, especially in a cell therapy context, would take place in SOPs, and including media that is standardized itself, such as Methicult, which may also be taken a step further with relying on automated quantitation, so to remove that human element from the quantitation of these assays. And in that case, it would be to use the STEMVision instrument. So as you can see, the 2 images on the right side of the slide there, STEMVision creates tiled high-resolution images of CFU assays in 35mm wells of a 6-well smart dish plate. And it looks at these images and analyzes them to determine colony subtypes for human CFU assays, and can also do total counts of mouse as well. In addition to quantitation, these high-quality images generated by STEMVision allow you to look at an entire plate to see what's going on, if there's any reason why that replicate should be excluded from analyses, etc. And really, you can zoom very close into the colonies and get great images of things that you normally are just sort of temporarily looking at under a microscope. And this data as well is actually very easy to be looked at years later when you're publishing, or emailed to collaborators around the world, or even when checking back into parameters that were related to a product that was developed and then used in later clinical trials. So in terms of validation of this instrument, we have internally validated STEMVision for counting CFU assays of human cord blood, mobilized peripheral blood, and bone marrow. And last summer, a study published by Vellier et al. Showcased their robust external validation of our instrument in a paper in Cytotherapy. So to finish off our talk today, here again is a brief overview of our genome editing workflow for CD34 cells in this case, from cell isolation to culture and analysis. And there's just a little short URL at the bottom there, which has the link to the full protocol, which includes guide RNA preparation and even electroporation settings. So to finish off this talk today, I really want to highlight the main takeaways that we just discussed. First, highlighting how important it is to consider every step of your protocols and the work that you're developing in the lab. And I hope you can see what I've shown today is just the amount of detail that has gone into the small considerations for how you treat your cells, especially when choosing a cell culture medium, even if it's supplemental and it won't be, the blockbuster part of your paper, it still really provides that firm base upon which your research takes place. And next, really to use the right assays to characterize your cells in your early research, as then these can be used as you move on to manufacture cell therapy products and into the clinic. And finally, to remember that whenever you're working with hematopoietic stem cells, you really are part of that history and are only a few steps away from the clinic yourself. So thank you everyone so much for your time and attention today. I hope this brief tour of our reagents for hematopoietic cell therapy research has been useful for you. If you have any more questions at all, please visit our booth, and also don't hesitate to reach out to me to discuss more. Thank you so much. Hello everyone. So now I'm going to stop the echo. I'm going to answer a couple questions that were submitted. So one question that came in was, how do iPSC-derived HSCs compare to bone marrow-derived HSCs in terms of when you're using the CFU assay? So CD34 positive cells derived from pluripotent stem cells are not functionally the same as CD34 cells derived from adult sources, though many people in the field are still working on this. Depending on the stage of embryogenesis from which the cells arise, their function at early stages of hematopoiesis are different. So, for example, the more primitive progenitors, as in early on in primitive hematopoiesis, they lack lymphoid potential because it's not needed at that point in embryonic development. And this is also seen in some iPS-derived hematopoietic cells as well. However, as myeloid and erythroid lineages are produced from iPSCs, you are actually able to do CFU assays, and we have some special methicult that's been designed to properly assay these cells. Sometimes the colonies may be a bit smaller, or certain lineages may look a little bit different. There may be issues with hemoglobinization of the colonies, and that's, as I said, why we have actually developed the media to properly visualize CFUs that are derived from iPS-derived hematopoietic cells. And let's just see another question coming in. Just to elaborate, so what does animal component free mean? How is this different from xeno-free or animal origin free? So animal component free media, so our STEMCELL ACF, contains only synthetic or recombinant proteins. So this is different from our xeno-free media, which would contain human-derived proteins, so human serum albumin. And in most cases, so animal component free, those recombinant proteins may be generated in an animal origin cell. So essentially purifying the cells from an animal cell where they were grown. However, animal origin free recombinant proteins are actually generated in plant or other cell types. So this is something that's actually quite interesting with our STEMCELL ACF media. It's still called STEMCELL ACF, but we've actually worked to create a product that is actually entirely animal origin free as well. So it's just that extra step of definition in terms of what's in the media. Let's see. And there is a question, so do you see large differences in outcomes between phenol red free and phenol red containing stem cell media? Of course, this is a great question because there is literature supporting the fact that phenol red can actually have estrogenic effects, which is one reason why we did choose to remove it from our upcoming GMP formulation. We have not extensively looked at the effect of phenol red having phenol red, including it in the media versus not including it. But these are studies that will be ongoing, and we will essentially, we chose to remove it just to remove any potential interfering components as we are designing STEMCELL ACF GMP as something that is more fit for the clinic. There's numerous phenol red free formulations that are out there and are widely used, so we wanted to kind of fit with what people are looking for. And let's see. Another question, do we have data for the engraftment of expanded cells that were cultured in STEMCELL? So this is another area where we're working on getting some of our own in-house data. As many of you know, these are quite intensive experiments, and if we want to do proper limiting dilution assays, there's many mice that are used. However, we are quite lucky. There's so many publications using STEMCELL, so this kind of extensive history where various groups have looked at culturing hematopoietic cells in STEMCELL and then transplanting them into NSG mouse models or other mouse models, it's out there in the publication record. And For STEMCELL ACF, an example in particular is a paper in 2018 published by Tavari et al. In Australia, and they were looking at the effect of different oxygen tensions when culturing their cells, and they did that in STEMCELL ACF and then showed that those cells did engraft. And maybe one last question. Oh, no, we're out of time. Thank you everyone so much for your time and your attention. And as I said, please send me an email or come chat at the booth if you have any more questions. Thank you.